This is a chromosomal analysis tutorial presented by SchrDbb. We reference ABC of Clinical Genetics by Helen Kingston. Hello, so here we're going to study chromosomal analysis. So chromosomal analysis, you have great disorders involving chromosomal analysis. Now, when you say chromosomal analysis, just to visualize the gross morphology of a chromosomes, and certain disorders such as Down syndrome, Turner syndrome, and Klinefelter syndrome are involved with chromosomal analysis. So now, this is like a chromosomal analysis done with karyotyping. So karyotyping, from here you can visualize the normal male chromosomes with a diagram demonstrating the G banding. So you need to know that we use the Giemster stain in order to visualize, to karyotype a chromosome, basically. And you need to know that you realize that every time that you have karyotyping of chromosomes, it is always in black and white. The question I'm going to ask you is, why is it that the karyotyping of chromosome is not in color but always in black and white? So the answer is simple. It's always in black and white because chromosome cannot be seen with light microscope. It can only be seen with electron microscope, and electron microscope do not have colors. So that's why when you use the Giemster stain in order to visualize chromosomes, we use the electron microscope, and always going to be black and white. So this is chromosome 1, which is going to exist in pair. This chromosome 2, chromosome 3, chromosome 4. So all that, these are the different chromosomes, and there are 23 pairs. At the 23rd pair, we have the X and the Y chromosomes. Is it clear? So using the Giemster stain for karyotyping. Now, next thing is to see cell division. So what is cell division? So you need to know that the normal chromosomes of the human body is made up of 46 chromosomes, and those 46 chromosomes are in pairs. So we have 23 pairs of chromosomes. So that 23 is usually considered as a haploid state of N. Now you need to know that cell division, the somatic cells, that is the cells that constitute the structure of the human body, are going to divide by mitosis. Is it clear? The somatic cells, the cells that constitute the structure of the human body, are going to divide by mitosis, except stem cells. So all of them with stem cells are going to divide by mitosis. Is it clear? Why the gonadal cells, the cells that are involved with reproduction, are going to divide by meiosis? Now, mitosis, the division by mitosis is going to produce the same number of chromosomes. The same number of chromosomes are going to be found in the mother and the daughter cell. Is it clear? Why in meiosis you are going to have half the number of the daughter, the chromosomes in the daughter cell compared to the mother cell. Is it clear? So that is the thing that is involved here. So this the thing which the division method which occur in the somatic cells is going to be by mitosis, while in the gonadal cells is going to be by meiosis. Now, mitosis is going to, you need to know that in the cell division phase, we mostly have two main phases. Is it clear? So we have interphase and we have cell division. Is it clear? So those are the two main phases inside the cell cycle. So the cell cycle mostly have two main phases. There is interphase and the cell division phase. Interphase is going to be the resting phase. Is it clear? So the resting phase is going to be divided into three. You have the G1, which is the first growth, which is called G1 here. So we have G1 during interphase. There is G1 phase, which is the first growth, and there you have the growth of the cell. The cell is having nutrients in order for it to grow. The second thing now is the S phase. S phase is the synthesis phase. Synthesis phase because here there is DNA synthesis. Is it clear? And then the next phase is the G2 phase. The G2 phase now is the second growth. In that second growth now, the cell is now mobilizing all the structure that is going to be involved now in cell division. Now, cell division can either occur in two ways. It can occur by mitosis or it can occur by meiosis. Mitosis and meiosis all have the same steps. The first step is prophase, and prophase is involved now with the condensation of the genetic material, the condensation of the chromosome. Now we have prometaphase and metaphase. In prometaphase and metaphase, the centrioles, which are attached to the microfilaments and the actin filaments, are going to with the myosin filaments, they are going to mobilize the chromosomes such that the chromosomes will align in the center. Is it clear? So the such that the homozygous pair of the chromosome align at the center. That occurs at the metaphase and prometaphase. Now in anaphase, we have now the movement, the separation of the homozygous chromosomes, or the separation of the different pair of chromatids, depending on if it is meiosis or mitosis respectively. Now the next one is telophase. Telophase now is you have the formation of different nuclei after separation. So when they separate, there is formation of different nuclei, and after telophase now we have cytokinesis, which is the division of the cells into two. Cytoplasm of the cells, the cell membrane into two. So all that is involved in cell division. Now this is what is involved now in meiosis and mitosis. You need to know that mostly so I said meiosis is mostly involved in gonadal cells, and there are mainly two gonadal cells. We have the egg cell or the ovum, and we have the sperm cell. So this is the diploid oocyte, and this is the diploid spermatocyte. So the first you have, when you have, this is actually this the primary spermatocyte here, and this is a primary. So the primary spermatocyte is going to have meiosis 1 to produce the secondary spermatocyte, and the secondary spermatocyte have meiosis 2 to produce the spermatid, and the spermatids now grow to produce the haploid cells. Here, this is the oocyte here. The oocyte here is going to divide by meiosis 1 to produce the first polar body, and then this next oocyte is going to still divide by meiosis 2 to produce the second polar body, and this one is going to be haploid exon. Is it clear? And the haploid exon with the haploid spermosome are going to fuse together. Since this is haploid having 23 pair, this haploid is having 20. This is haploid having 23 chromosomes, and this is haploid having 23 chromosomes. All of them is going to bind to produce a 23 pair of chromosomes. That is 46 chromosomes, such that the genetic component of the offspring stay the same as that of their parents. So this is how genetic representation occurs in mitosis. So in mitosis, you're going to have alignment of the different mother and father chromosomes in the center, and then the pair of chromatids are going to be separated, such that all the cells are having the same content as the parent cells. Now in meiosis, you need to know that what meiosis is divided into meiosis 1 and meiosis 2, and the characteristic of meiosis is that you have change in genes, like you have crossing over. So what happened in meiosis is that what you first have meiosis 1. So meiosis 1 occur where you have the parents, so the parent genes are going to be at the center, and then they are going to be separated. They are going to be separated following crossing over, when there is a change of gene from one mother chromosome to the one father chromosome in the child. Is it clear? So now in the homozygous pair are going to be separated in the meiosis 1, and this homozygous pair separation is from this point that there is the division in the chromosome number from 46 to 23 chromosomes, and then the chromatids now are going, the sister chromatids are going to separate now into two in order for it to enter into the different gametes that are going to be produced. This one is going to be characteristic of meiosis 2. Now chromosomal analysis. Now we need to know that before you have we go to chromosomal analysis, you're going to have definition of certain terms. When you say euploidy is just meaning normal set of chromosomes, and it's meaning that it is 2n, 23 pair of chromosomes. That's euploidy. Now polyploidy is when I say I have a disorder which is a polyploidy, it is a multiple set of the normal set of chromosomes. Is it clear? A multiple set of chromosomes. So when I have 3n, 4n, 5n, those are polyploidy. Example, if I have a triploidy, it means that I have 3 pairs of 23 chromosomes. Is it clear? It means that I have 23 times 3, which is equals to 69. I have 69 chromosomes. So that's a triploidy. Is it clear? Tetraploidy means that we have 4 pairs of chromosomes, of the normal set of chromosomes. So it means I have 4n. So it means that it's 23 times 4. Now aneuploidy is an addition of one chromosome number. Is it clear? So that is what is called aneuploidy. An addition or subtraction of one chromosome number. It is not a whole set, but it's only one chromosome number. Example, when you have the addition of chromosome 21, is it clear? It is called trisomy 21, and this is the same as Down syndrome. If you have a subtraction of chromosome X, is it clear? It in a female, it is going to be called a X0. So it is going to be called a Turner syndrome, and this is a monosomy. So mosaicism. So in mosaicism, what happens is that certain cells are going to have a normal genetic constitution, while other cells are going to have an abnormal genetic constitution because of a somatic mutation. So here in mosaicism, what happens is that we have 2 different cell lines. One is going to be normal and another is going to have an abnormal cell line. Why? Because there was an abnormal cell division that occurred in the precursor of the cell line. Is it clear? Example, you have a 46XX, this is a female with certain cell lines having 46XI, while other cells line are having a Turner syndrome mosaicism. Is it clear? And the next one now is chimera. A chimera is the presence of 2 different cell lines derived from a fusion of 2 zygotes. So this one is from one zygote. The difference between mosaicism and chimera is that mosaicism is from one zygote. So the presence of 2 different set of chromosomes, cells line from one zygote, while chimera is the presence of 2 different cell lines from the fusion of 2 chromosomes. Example, one may be 46XX, another may be 46XY. Is it clear? So that's the difference. You have 46XX, you have 46XY. So this is a true hermaphrodite. We have fusion of 2 zygotes together. Is it clear? So it's going to go a different cell line. Now karyotyping. So this is actually karyotyping, as you can see with the GMSA stain on a electron microscopy. So in karyotyping, you need to know that the center here, the small part is going to be called the P arm, while the long part is going to be called the Q arm. Is it clear? Now the small part has only one portion, which is 1, and the long part has 1 and 2. And the 1, so this 1, 2, and 1 and 2 are called the bands. And now here are the sub bands underneath here. Are the sub bands. So we have 1.1, 1.2, you have 2.1, 2.2, and all that. So these are the sub bands here. And this show the different locus of the genes. So one gene can be located here, another gene can be located here, another gene can be located here. These are the different locus or location of the genes. Is it clear? It's very important. So these are certain disorders which are involved in these disorders. So aneuploidy is an example. You have trisomy 21 like Turner syndrome. Monosomy X chromosome is aneuploidy. Trisomy 21 is Down syndrome. Monosomy is Turner syndrome. 47 chromosomes XXY, it is Klinefelter syndrome. So if you have structural abnormalities, so these are chromosomal abnormalities that you can have. Chromosomal abnormality, first chromosomal abnormality is a polyploidy, a numerical abnormality. Is it clear? So a numerical abnormality is just a change in the number of the chromosomes. Polyploidy is one where you have an increase in, or you have a polyploidy is an increase in the set of chromosomes. So example, you have triploidy where you have 3N chromosomes, you have 69 chromosomes, it's lethal. Aneuploidy, you have trisomy 21, which is Down syndrome. You can have monosomy X chromosome, it's Turner syndrome. You have Klinefelter syndrome, XXY. Now, structural abnormalities of chromosomes, which can be diagnosed with chromosomal abnormalities. We have deletions. So deletions, example, if you have a terminal deletion of the 5p arm, I said that p is short arm. If you have terminal deletion of the short arm of chromosome 5, it is going to be called the Cri-du-Chat syndrome. Is it clear? If you have an interstitial, so you see, these are the different forms of deletion. Deletion can be divided into terminal deletion and it can be interstitial deletion. Interstitial deletion means that it is in between, while terminal deletion means that it's at the end. So it is deleting the part of that locus of that gene at the end, while interstitial is deleting that part of that gene at the middle. So if you have an interstitial deletion of the short arm of chromosome 11, is going to be associated with Wilms tumor. So Wilms tumor is generally a tumor which is found inside the urinary system, mostly in the kidney, present at the level of the newborns. Very important tumor inside neonates. Inversion. So inversion is another disorder. So if you have a pericentric inversion, inversion can be divided into pericentric and paracentric. Is it clear? Pericentric inversion, it means that inversion at the level of the centromere. Is it clear? While paracentric is far away, or terminal inversion is far away from the center. So you have a normal phenotype. A normal phenotype is going to have a pericentric inversion. So you have no change. Usually you have a normal phenotype when you have no removal of a gene or no addition of a gene. Is it clear? So that's when you have a normal phenotype. So if there is just inversion, there is translocation, and it is balanced, usually the phenotype is normal. But their future generation is going to be abnormal because when the normal assortment is going to occur during meiosis, and homozygous pairs are going to separate. That's when their children is going to have the different disorders. So duplication. In duplication here now you have example like the isochromosome X. So we have a fusion of the long arm with the loss of the short arm of chromosomes. So here you have infertility in females, duplication. You have also a ring chromosome like ring chromosome 18. We have mental retardation syndrome. We have fragile site like fragile X chromosome. You have mental retardation syndrome. Translocation. Now translocation can be a reciprocal translocation or it can be a Robertsonian translocation. In reciprocal translocation, there is a transfer from a chromosome, so it is like a crossing over between two non-homozygous pairs. It is just the definition of a translocation. Translocation can be discovered, it can be described as an exchange in chromosome between two non-homozygous pairs. It can be balanced or unbalanced. When it is balanced, it means that what? Let's say that the chromosome 14 was having a certain gene, it transfers to chromosome 21. Is it clear? And 21 transfer that same gene to chromosome 14. It is going to be said to be balanced. That patient is not going to have any phenotypic disorder, but the children of that child is going to have the abnormality. So this is a balanced reciprocal translocation. You may also have unbalanced reciprocal translocation if 14 transfer but 21 does not transfer. Now in Robertsonian translocation, it occurs mostly between acrosomes. And what are acrosomes? Acrosomes are chromosomes that have a very short p arm. So when there is a transfer from the short p arm of one chromosome to the short p arm of another chromosome, it's the p arms are actually very short though, so that's why they actually fuse together. Is it clear? Example, chromosome 9 and 22, which produces a Philadelphia chromosome producing the chronic lymphoblastic leukemia, is one example in a Robertsonian translocation. So the father is going to have a risk of chronic lymphoblastic leukemia, and the children are going to suffer from the disease, like certain aneuploidy or triploidies. So that is what is involved here. So you also have physical and mental handicap when you have a 14-13 chromosomal abnormality. So it's mostly occurring between acrosomes, because you have three types of chromosomes. You have acrosomes, you have merosomes, and you have telosomes. So those are the three main types of chromosomes. Acrosomes are chromosomes that the P arm are very small compared to the Q arm. So the P arm are very small and they are minute, so that they cannot even be measured. Now telosomes are one that the P arm is shorter than the Q arm, while merosomes are others that the P arm is equal to the Q arm. So there are certain chromosomes that have those genetic constraints. So this is reporting a karyotyping. So when you do a karyotyping, how do you report? So to report a karyotyping, this is a normal female. So a normal male is going to be 46XY. Is it clear? Now this is now a male now with Klinefelter syndrome, 47XXY. This is a male with the triple X syndrome. So this is when the total number. So this is total number. You first start with this total number, then you put the if it is XX or XY or XXY. So this is how you first start. Now additionally, to see if there's a 14XY plus 23, if there's a trisomy 21, you put plus 21 like that to show trisomy 21. If there is an addition of the 12p, the short segment of chromosome 12, you see that the short segment of chromosome 12 is lengthened more than the normal. So that is additional unidentified material, the short arm of chromosome 12. Is it clear? So that's an additional component. Now if you want now to show that there are two different cell lines, let's say one cell line and there's another cell line. There's one cell line which is a normal female portion, and there's another cell line which is having trisomy 21. You say that it is a Down syndrome mosaicism. Is it clear? If there is one cell line which is normal, another cell line having triple X syndrome, another cell line is having Turner syndrome, it is a Turner triple X syndrome mosaic or mosaicism. Now structural rearrangement. So if there is structural rearrangement like deletion, so for deletion you just see the deletion of the chromosome 11 P, the band is the band of is a 13 band of the short segment of chromosome 11. So that's how it's writing, del 11p13. So if there is the next one is 46XX. So when you want to show translocation means that it's transferred from one to another. So it is a translocation of chromosome 7. So there is the interstitial translocation, means that what chromosome 7, there's a transfer from chromosome X to chromosome 7, meaning that what the X has transferred the band 21 of the short segment of chromosome X has been transferred to the long arm, the band 23 of the long arm of chromosome 7. So that's the translocation. So that's just saying that there's translocation between chromosome X and 7 with the breakpoints which are this. So this is molecular genetic cytogenetics. So in molecular cytogenetics, you can use now what is called fluorescent in situ hybridization method. So here you see that what you see, this is fluorescent in situ hybridization. What happens is that in that technique, you just coat a particular gene with fluorescent substances, such that when you take a microscope, it can glow. Let's say example, I have a particular gene. I want to find if there is a genetic disease. Is it clear? You need to know that the fluorescent gene is going. Let's say that I create a fluorescent gene that pairs with an allele which is an allele for a genetic disease. So in fluorescent in situ hybridization, if I visualize that there is the fluorescence of that gene that I have created, basically it means that the patient is going to have the allele of that complementary pair of the gene that I created, basically. So like in this case, we have fluorescent in situ hybridization of a normal metaphase chromosome hybridized with chromosome 20 probe derived from whole chromosome. So this is another case. Fluorescent in situ hybridization, metaphase chromosome of a male 46XX chromosome hybridized with a separate probe, both X and Y chromosome. So you see have a probe of X and Y chromosome. The X chromosome probe has been hybridized to both X chromosome, basically, and the Y chromosome probe has been hybridized to the Y chromosome. So we have now frequency of the abnormality in spontaneous abortion and stillbirth. So usually spontaneous abortion, you see that all spontaneous abortion which occur, some 50% of all spontaneous abortion is going to have chromosomal abnormality. So you need to know that most 50% of all the spontaneous abortion, 50% of all the spontaneous abortion is going to have chromosomal abnormality, and that chromosomal abnormality, most of them is before 12 weeks of gestation, because at before 12 weeks of gestation, you have 60% are going to be having chromosomal abnormality. So if a woman is having spontaneous abortion during the first trimester, that is before 12 weeks of gestation, do not prevent her from having the spontaneous abortion, because that child is usually going to be abnormal during the first trimester. Now, in the second trimester, that is from 12 weeks even to 20 weeks of gestation, you see that from this state now, it is only 20% is going to have chromosomal abnormality. So you can prevent the child from going out by doing cervical cerclage. And now in stillbirth, when stillbirth, it means that what is an intrauterine growth restriction, means that the fetus die inside the mother, inside the uterus. So you see that only 5% have chromosomal abnormality when they are already in the third trimester. They die in utero. So when you want to define stillbirth is not like missed abortion. Missed abortion is intrauterine death before viability, while stillbirth is intrauterine death after viability. Now, frequency of chromosomal abnormality in newborn infants. See, all newborn infants, only 0.91%. Autosomal trisomies, constituting of autosomal trisomies, only 0.14. Most of them is going to be autosomal rearrangement, like balance translocation 0.5. Unbalance is going to be less, and sex chromosomal abnormality is going to be less. So the two major things that are going to be inside newborns, we have balanced translocations, and we have you have balanced translocation, you may have autosomal trisomies. So from here we are finished with the tutorial. I'm saying thanks for your kind attention. Don't forget to like and subscribe for our channel.